Hi, everyone. Today's topic is protein structure determination with cryo-EM. Cryo-EM as an experimental technique has existed for over half a century, but it's really been growing in popularity probably over the past decade, both amongst biologists who want to push forward discovery, but also amongst computationalists who are faced with new challenges in processing the data. And so I'm really excited to be giving this intro on what the method's about, and then in about an hour, Ellen will pick up and talk about how our research group is actually implementing tools to process this sort of data. So as a starting point, as we all know, proteins are really fundamental to all the processes that go on in our body, from catalyzing reactions to being the building blocks of our bodies to transmitting signals. And so understanding proteins is really fundamental, not only to understanding life, but also intervening rationally within medicine and health, and also driving innovation in industries like nanotech and biotechnology. There are many different lenses through which we can study proteins, but today we're specifically interested in studying proteins through the lens of structure. So proteins take on really a wide range of structures, and just as importantly, proteins are also highly dynamic. So they're flexible, subunits can bind, unbind, and this dynamic structure is really critical to their function across multiple scales. So on the left here, you can see that these sort of really local fluctuations of side chains within these binding pockets drive binding to substrates. In this dining molecule, global movements of domains allows this protein to carry cargo across the cell. And on the very right is the nuclear pore complex, which consists of hundreds of different protein subunits, which are interacting in concert to selectively allow passage of substrates into and out of the nucleus. And so there are multiple techniques we can use to study this structure. They broadly fall into three categories. One is experiment, including cryo-EM, crystallography, NMR, where we use experimental techniques to directly probe samples of proteins. Physics-based methods like molecular dynamics, where we use our knowledge of biophysics to simulate the dynamics of proteins. And finally, statistical methods like AlphaFold, which use these data-driven priors to make sort of plausible hypotheses about structures from their sequence. Today, again, we're specifically focusing on cryo-electron microscopy, which is an experimental technique. Again, it's been around for a while, but currently it's going what is sometimes called a resolution revolution, where due to advances both on the hardware side in the microscopes, as well as on the software side in terms of processing algorithms, we're now able to process a much broader range of proteins and also achieve really high resolution structures, like you can see on the right. This also comes with a whole new host of computational challenges and opportunities. And so the plan for today, I'll start off with an overview of the whole cryo-EM pipeline, going from, if you're a structural biologist interested in studying some protein, how do you go from an idea of that target protein to actually depositing a structure in a database like the Protein Data Bank? And then I'll spend some time talking about the classical algorithms for cryo-EM reconstruction, just to get the underlying theory. And then that'll set up the final part of my talk on modern methods for heterogeneous reconstruction. And then finally leading into Ellen's talk, giving some broader context for these methods and some of the newer developments within our lab tackling these challenges. So starting out with an overview of the pipeline, the first step is that given a target protein, I'm first going to amplify and purify that protein from my cells and flash freeze it or vitrify it under liquid nitrogen conditions. So this freezing process, it traps the molecules in place so that we can image them under long exposures. And it also increases their tolerance to radiation damage within the microscope. One other important aspect of this freezing process is that, for context, one of the other major experimental techniques for structure determination is X-ray crystallography. And in crystallography, you typically have to crystallize the sample. And that's often a very difficult thing, especially when you have highly flexible molecules, really large complexes consisting of multiple subunits. And so cryo, first of all, has really allowed access to a broader range of protein specimens because more samples are amenable to just flash freezing than they are to crystallization.
Yeah, I think I might ask some basic questions during your talk because it's outside of my field, but I find them fascinating. So for X-ray crystallography, I think I've heard of it in context of individual proteins, like proteins being used for proteins to determine structure. But is it done on a cell level like cryo-EM is? Or am I confusing it with something else?
Sorry, could you repeat on the what level?
So with cryo-EM, you take cell level images, right? Do you get that for X-ray crystallography as well? Or is it like for molecules,
Yeah,
not the whole like distribution of, different proteins in the cell?
Yeah, so I guess in both, for the cryo-EM that I'm presenting here, it also is a similar setup as crystallography, where we're not imaging a cell, we are just purifying a specific protein and imaging that sample. There's an extension of cryo-EM for this in situ cellular samples, which I'll talk about.
Okay, I see. Thank you.
Yep. Yeah, so we freeze our sample, and the other really important aspect of this freezing process is that because the freezing process is so fast, we're trapping these molecules in sort of a native state, where the conformations of each of those individual particles in the sample will be approximately distributed according to the sort of thermodynamic equilibrium. And so that provides us with a really powerful opportunity to study not just the single sort of average static structure, but actually access this full conformational landscape. So once we've gotten this sample, we can put it into an electron microscope, which looks like the thing on the left, and irradiate it with electrons. And so what the microscope gives us is a micrograph, or a collection of micrographs, which is a 2D projection of the electron scattering potential, the sample. The key idea is that you have water, and then you have these particles embedded within them. And so due to differential scattering, you end up getting sort of the contrast in these images. So in the micrograph, each one of those dark spots you see is one individual molecule of the protein. And so in order to do structure processing, the first thing we have to do is actually extract each one of those individual particles. This can be done in a variety of ways, including machine learning techniques or using prior structural templates. But after extracting those particles, what we end up with is usually between 10,000 up to maybe even 10 million of these individual 2D images. And each one of those consists of a unique particle captured from the sample. One challenge in general in cryo-EM data sets is that there tends to be a lot of junk or outlier particles. This is both due to imperfections in the purification and freezing process, as well as inaccuracies in the particle picking process. Yes.
Oh, can I ask you, thank you. Can I ask you a quick question about the electron microgram? So I know like cryo-EM kind of took off a lot in the last like maybe 10 or 15 years. And I heard one of the reasons was like just imaging got better or the cameras got better. Is that correct, or am I imagining that?
Yeah, that I don't know the exact year, but yeah, direct electron detectors where you can get direct counts of electrons spatially, gave much better sort of signal in these images. I don't know if you have additional context, but yeah.
Just echoing Rish, like better hardware increasing the amount of signal in the collected images such that now you can get atomic resolution structures. And this was about like 2013.
Yeah. So characteristically, these data sets do contain a lot of junk particles. And so this is going to be a processing challenge that comes up later in Ellen's results again. But at this phase at the 2D level, there are certain things we can do, specifically sort of grouping similar looking images and sort of throwing out classes that correspond to junk. And finally, the core computational question that we're interested in is the 3D reconstruction problem of how do we go from all these really noisy 2D projection images of these particles viewed at different angles and go to the underlying 3D structure or 3D electron scattering map. This is in general a very challenging problem, both because, as you can see, the images are very noisy. The signal to noise ratios can be as high as 0.1, but also as low as 0.001. So sort of signal to noise ratio of sort of 1 to 1,000. Also, the particle orientations are unknown. And so, in the sample, all those individual molecules are rotated randomly. And so in order to sort of combine the signal, we need to be able to have a common reference frame that we can average that signal. And so the second thing is that we don't know what orientation each of those particles is in. And so we're going to have to estimate those parameters in addition to the underlying structure. Another challenge, but also a major opportunity, as I said, is structural heterogeneity. So each one of those images, again, corresponds to one completely unique molecule. And so this gives us the ability, instead of reconstructing a single average structure, to reconstruct a full distribution of structures. Again, that's a major opportunity that allows us to understand more about biology, but also presents challenges. And then finally, after usually long processing pipelines, if you achieve a sufficiently high resolution 3D reconstruction, you can then use that to guide building an atomic model, which is typically what gets deposited in a database, such as the protein data. Okay, so that's a lightning overview of the cryo-EM processing pipeline. Next, I'll talk about how this reconstruction problem of 2D images to 3D structure has traditionally been tackled.
Quick question.
Yeah.
Can you briefly address the difference between cryo-EM and cryo-ET?
Sure, yeah. Actually, yeah, this is a good slide to explain that on. So in cryo-EM, so there's your sample, and we acquire just one micrograph, one image. In cryo-ET, we then tilt the sample and acquire multiple different views of the same sample. And so you can essentially treat cryo-ET stands for tomography, so you can treat cryo-ET as a multi-view version of cryo-EM. And the reason why that's helpful is because, especially when you want to study structure within the cellular context, so you want to look at these molecules in the context of membranes and organelles and other aspects of the cell, you typically want a full 3D reconstruction of the sample containing all those different organelles. And so yeah, that's a situation where tomography can be helpful, where you acquire multiple views at different angles, and then you can do a back projection to get a full tomographic volume.
So I guess for that one, you wouldn't go about with purification. So this one, you would purify the protein before you do cryo-EM, and there you would just use like a cell or a tissue directly?
Yeah, exactly. Yeah. Yep. So before talking about reconstruction, we first need to build a model of how these images are formed. And so this equation here is showing that, so let V be the underlying structure. So here I'm just assuming homogeneity. So we just have one single structure that doesn't have any conformational changes. So we're going to call that structure V. Any one of these images is obtained by projecting that volume along some direction, some angle. That angle I'm going to call phi I, and P is just the projection operator, essentially integrating or summing across that volume, so projection. That image is then corrupted with the contrast transfer function, which describes the aberrations of the microscope. And then finally, we add Gaussian noise. This noise corresponds to shock noise of the microscope detector, sort of owing to the quantum random nature of electrons. And so that forms one image, which we denote by xi. Another point, actually, this phi I, this orientation or this pose I talked about, actually consists of two components. So one is a random orientation of this molecule within the sample. The second is a 2D translation, because when we do our particle picking, typically without a 3D reference, the particles within those 2D images are going to be a little off-center. So we both need to account for this random orientation as well as this 2D translation. So then the cryo-EM reconstruction problem is the inverse of that image formation model, where what's observed is a data set of these xis, these images, and what we aim to find is the underlying map V. A note that just in what follows, I'm going to assume that those contrast transfer function values C are known and fixed. That's a reasonable assumption because you can estimate those parameters typically earlier in processing in the 2D stage. However, again, the poses are unknown. And so that is something we're going to have to estimate. Yeah.
I guess for the Gaussian noise model, is this is this actually backed by the data, as in if I run it on something blank, I get out Gaussian noise, or is this just to make it tractable and easy to solve post fact?
In terms of being physically realistic, there are more realistic models. So it is shot or more like Poissonian noise, and also the forward model. Even in the forward model, treating it as a just straight projection is usually a reasonable assumption, but you can have even more expressive models where you do like multi-slice wave propagation, and yeah.
So sorry, so should I think of this as like counts of how many times I'm like my imaging modality kind of went through and hit the sensor underneath?
Yes, yeah.
So it's fully a positive kind of thing that I'm observing?
Yes, it is an electron count, yeah.
Interesting, okay, thanks.
Yes.
Hi, sorry, one more question. Why do you, like, if you're automatically segmenting out the particles, like, why wouldn't you just assume that they were centered? Like, if you're just, yeah.
Yeah, so the particles will be centered with respect to the image that they're in. But when you need to combine all these images into a 3D reconstruction, you then are going to have to sort of shift those images a little. Without a 3D reference, the notion of a center for a 2D image is ill-defined. Yeah. Okay. So this is the problem we're interested in. Again, so we're observing the x's, these images, and we aim to recover the underlying structure. We can think of this in a probabilistic framework. And so we can frame the reconstruction problem as finding a set of parameters, so the structures and these orientations, that maximizes the likelihood of the data, given the parameters. Each one of those images is independent. So the full likelihood is just going to be the product of the individual likelihoods of observing each of those images. One sort of assumption we can make at this phase is the thing we really care about is optimizing that volume V, and those particle orientations or poses are just nuisance parameters that we don't directly care about. And so we can marginalize over all those poses. The marginalization is over the space of poses, which consists of an element of SO3, so rotations, and an element of R2, which is a 2D. Okay, so from this, all we need is really an expression for this. So what is the likelihood of a single image given a volume and a pose? To get that, we can think back to our image formation model. So again, we said an image is formed by projecting this volume and then adding some Gaussian noise. Alternatively, we can reformulate that by saying that the image itself is a sample from some Gaussian, multivariate Gaussian distribution centered on that clean projection image. With variance sigma. And so given that interpretation of the image, the likelihood of observing the image given the model parameters is just going to be what you get when you plug the image to the Gaussian PDF for the multivariate Gaussian centered on this mean, which is the clean image, and with that sigma. And so, if you recall sort of the Gaussian PDF, this takes a form like this. It's sort of the L2 norm between the point and the mean with this normalization constant out front. Okay, sorry. Okay, so we have this expression for the full likelihood. We have this expression for the likelihood of a single image. So all we have to do is then take this and plug it into there.
Oh, can I ask a question?
Yeah.
You said that like the protein itself is in a range of conformations. So is the V also like a distribution, the cryo-EM density?
Oh yeah, so in what I'm showing here, I'm assuming that this is a protein that is static. So we just have one underlying structure. I'll sort of build up to the case of having, that full distribution. So we can take that, the single image likelihood, plug it into the total likelihood, you get an expression like that. And then the optimization problem essentially ends up being maximizing that full likelihood with respect to the volume V, and you get an expression like that. So again, this expression is saying that we want to find the volume V that maximizes the likelihood over all the images, again, marginalizing over the poses. Unfortunately, this expression is intractable. We won't be able to solve for V in a closed form solution. And so we'll need another technique. So the way this is typically done is expectation maximization, which is a very common approach in these sort of problems. And the idea is that the marginalized full log likelihood that we just computed is intractable, unfortunately. But we can actually optimize a different quantity instead, which is this expected log likelihood. You can read more on the evidence lower bound or elbow, but the basic idea is that this objective, it's not the same as the true objective I showed, but it's a lower bound on it. And by optimizing this, we can get a reasonably good optimization of the true objective as well. If we take a look at this quantity that we want to optimize, there are two ways to sort of maximize this, right? One, so this is saying this is an expectation of this log likelihood over the space of poses. And so to maximize this, there are essentially two things we can do. One is to draw these poses from a distribution that matches the true distribution of poses. The second thing we can do is choosing a volume that maximizes this expectation. And so the EM algorithm is essentially alternating between these two steps. In the E step, what we're doing is estimating the best, estimating the likelihood of different poses. So first of all, okay, recall that the pose space is continuous, right? Both translations and rotations. In order to make things easier for us, we're going to discretize it into sort of a set of candidate poses that we want to evaluate. And so this is saying that in the E step, for each one of those images, we're going to evaluate how likely is that image to arise from each of those candidate poses. And so we essentially get a posterior distribution of again, how likely each pose is for that image. And then the M step is, given those estimates of the poses, then choosing a volume that maximizes that expectation. So again, key idea, we're just going to alternate between A, optimizing the orientations or poses, and then optimizing the volume given those orientations. Luckily, this is sort of a complex equation, but the key takeaway is that this form of the objective does have a closed form solution. So by going from that original objective to this lower bound, what we end up with is an explicit expression for the volume that maximizes that objective. And if you try to interpret what this is saying, it's saying that the optimal volume is essentially in 3D space, taking all your images and basically taking a weighted sum over them. So every image for every single orientation, taking this weighted sum where the weights are higher for orientations that are sort of more likely to produce that image. Just to make this more intuitive, here's a graphic. So again, the E step consists of estimating the best orientation parameters given the current volume. So here's, let's say, our current volume. We can create a bunch of, we can use the forward simulation model to create a bunch of clean projection images for each of those candidate poses. And then we can compare each of those projection images against the true image and compute the error between them. This will give us a sense of which ones of those candidate poses were good poses, which ones of those were likely, and which of those is unlikely. And so this gives us, that reprojection error gives us these weights of, what poses are the most likely. So given those estimates of the poses, the M step is then essentially back projecting all those images according to the poses. When I say back projection, you can think of it as the inverse of the projection operator, right? So for projection, we took this volume and we essentially integrated or summed, we did a projection. back projection essentially corresponds to doing the opposite process, taking this 2D and going back into 3D. What does that actually mean? I think it's easier to interpret in Fourier space. So far, I've been talking about the real physical space. Fourier space is the space of spatial frequencies. And so in this space, according to something called the Fourier slice theorem, in real space, again, you have a volume, you project it, you get a 2D image. It turns out in Fourier space that the Fourier transform of an image is a central slice of the Fourier transform of the volume. So the image is just the slice of the Fourier volume. And so that's what we mean when we say back projection. When we say we back project an image according to some pose, it means we're just going to insert that image into the 3D volume along some orientation in Fourier space. And this corresponds to the inverse of the forward operation. So again, just to recap, E step, estimate the best poses. M step, according to those best poses, basically place all these images back and do a back projection in order to recover the underlying volume and keep iterating, going back and forth between these two steps. So this is like a very general framework for how to do reconstruction. It's not even specific to cryo. This is sort of a general way of formulating any sort of tomographic reconstruction problem. Specifically in cryo, there are some remaining challenges which prevent that procedure from being effective. And that's one just property of the expectation maximization algorithm is that it always improves the likelihood, but that also means that it will probably get stuck in a local optima unless you initialize it properly. Second, as we see, these images are extremely noisy. And so during this process of aligning images to the volume, it's very easy to overfit to the noise in the images rather than the underlying signal. And so we need some strategies for dealing with both these problems. For dealing with the first problem of how do we deal with initialization so that EM actually converges, one innovation introduced around 2017 by this software package CryoSpark was to, before directly running the expectation maximization algorithm on a completely random initialization of the volume, they first do gradient descent on that true objective. So this was the true objective that we initially derived. Even though we don't have a closed form solution for it, we can do gradient descent on it. And so they start out just doing gradient descent to optimize the volume with respect to that true objective. That gives you not a great volume, but a reasonable initialization, which you can then feed to the EM algorithm to increase the chances of actually finding that global minimum. The second innovation pertaining to how do we prevent overfitting our parameters to the noise in the images. This was introduced by Relion, which is a different software package. And the idea is that the objective I presented was the maximum likelihood estimator. But you can also formulate this in a Bayesian framework and do a maximum a posteriori objective. So the map estimator is going to have the MLE objective along with this prior term, which serves as regularization. That prior term is typically just saying that each of the Fourier components, so each of those, the powers of each of the spatial frequencies within the volume is drawn from a zero mean Gaussian. It's a relatively simple prior, but the variance of that prior is usually also optimized during this process. And the idea is that we can sort of estimate reasonable frequency dependent variances depending on our sort of known knowledge of the power spectrum of cryo-EM images. So this essentially just provides a form of regularization where especially early in training, when the amount of information content at those high frequencies is quite low, this regularization sort of prevents overfitting to those high frequencies and sort of dampens those components early on in the process. Okay, so that's sort of the EM algorithm. Question was just asked on how do we actually discretize the poses. So again, the specific thing we're interested in is, again, I presented this way of optimizing the volumes. And I said we can take that integration over the entire space of poses and turn it into a sum over a discrete set of candidate poses. So one question is, how do we actually discretize the space? So we can think about how to do it. What if we just brute force it, right? So what if we take the space of poses and just break it up into a really fine grid? That's not going to work, because if you look at how many poses we'd have to get 1 degree of rotation error, it would correspond to nearly 19 million different poses. And you can do some back of the envelope math. If you have if your images are of size 128 by 128, even for a single image, computing like a reprojection error or doing a back projection, it's going to be on the order of 310 billion operations. Now we have hundreds of thousands or millions of images, larger images, and we need to run EM in an iterative manner. And so this just becomes completely intractable. And so we need a strategy for discretizing the space better. And so we're going to do 2 things. And they're both sort of based on this observation that in general, the error between the pose error is smooth with respect to the pose, meaning it doesn't jump around a lot, right? This is the optimal pose. As I move away from the pose, the error sort of falls off gradually. So we can make use of this observation to do a couple of heuristics. And again, so they're based on this idea, again, that we have large areas of pose space that are either good or bad, so we can reject large areas at a time. The first idea is subdivision or hierarchical grid search, which says, we can start out with a very coarse sampling of poses, so split up the space very coarsely, find the best poses within this coarse space, only take the best ones, divide them up even more, search again, take the best ones, divide them further, search again, and essentially you only evaluate the really fine samplings for just a few poses instead of all of them. So that's one idea. The second idea is that when we compute that pose error, that reprojection error between this clean image and our real image, so again, that's that error, right? So X is the real image, CPV is that reprojection, and taking the L2 norm gives us a measure of the error between those 2 things, a measure of how good that pose is. We note that this is just summing over all the pixels, right? So just compute the error pixel by pixel, sum over all of them, which means we can also split it up into 2 terms, one corresponding to just low frequency components of the image, and one corresponding to the high frequency components of the image. And now one key observation we can make is that typically this pose estimation process aligning these images is dominated by the low frequency components. And so we can actually ignore those higher frequency terms and only compute the error on the low frequency terms. And so that is cheaper, right? Because we're only computing it on a subset of the frequencies, but it also actually improves the alignment because it avoids, again, sort of aligning to the higher frequency noise components. And in practice, one question is like, how do you actually choose what constitutes low frequency? Typically, the bandwidth is increased over time. So very early on training, when you don't have a good volume, you just have a blob, you use a very low value of L, meaning you only evaluate these errors on the very low frequencies. And as you progress in training, you just keep increasing to include higher and higher frequency components as you build up this higher resolution reconstruction. One last note is that I've sort of presented all that in terms of SO3, so rotations. But like I said, we do also have to estimate these 2D translational shifts. And doing so, we can use very similar techniques. As I said, for the rotations, in particular, we can still do this sort of hierarchical grid search, discretizing the space of 2D translations. One other note I'll make is that when I presented the EM algorithm, I presented in a sort of a purely probabilistic framework where we essentially assigned a probability to each possible pose for each possible image. What's done in practice in a lot of programs is that instead of constructing that full posterior over poses, you actually just take the best pose. And so you alternate between estimating the absolute best pose and then using the best pose to then update your volume reconstructions. Okay, and then finally, So again, everything I presented was through the lens of homogeneous reconstruction, so assuming we just have one single underlying static structure. But now we can start thinking about how do we incorporate the structural heterogeneity where you have multiple underlying structures. So we can modify our image formation model. So assume we have a setting like this. So here is a ribosome, and this is an assembling ribosome. So essentially, as the ribosome is maturing, different subunits get added on. And so here we have this case of discrete heterogeneity, where each of those images may arise from one of, let's say, those 4 structures. So how do we account for that, where we want to reconstruct all 4 of those structures from our imaging data? We can modify our image formation model. So now it's going to look extremely similar, except instead of having just a single V, I have this V indexed by AI, where AI is the class of that particular image. So now the multi-class reconstruction problem is, again, instead of finding a single volume, find the entire set of optimal volumes, assuming that we have K volumes given the data. Again, the challenge here is that all we have is this raw 2D image data set. We don't have labels on each image of which structure it arose from. So we'll have to both find the volume, but then also find those class assignments of which particle belongs to which volume. I won't go through the full derivation here. This is just to show that the objective looks very similar to what we saw in the homogeneous case. So I'm assuming here that we know the poses ahead of time. That's actually a reasonable assumption. Sometimes you do the sort of 3D classification after doing an initial homogeneous reconstruction. So with that assumption, we saw in the original objective, we were essentially marginalizing over the space of poses. Here, we're just marginalizing over the class assignments. Okay, so that's the only thing that really changes in the objective. And the EM algorithm is going to look very similar. Again, recall, in what I just presented, we alternated between estimating the poses and updating these volumes. We're going to do the same thing here. We're going to, in the E step, we're going to instead estimate the class assignments or the probabilities of an image belonging to different classes. And then using that in the M step, we're again going to update the volume, where if you look at the update equation, the volume for a given class is going to be, again, this sort of weighted average over every image, weighted by how likely is it that that image belongs to that class. Yeah.
Every class, we still have different possible poses though, right?
Yes, that is true. So it is, right, in a full formulation, there is a per class pose as well. It sort of depends on the route you take during processing. So there's one route, do a homogeneous first, just use those poses, purely classify. There are other routes where you can do the joint optimization. So yeah, so the process is going to look very similar, just doing this optimization over class assignments. In practice, 3D classification, which I just showed, is still is quite challenging for a multitude of reasons. First, we assume that each image arises from a single, from one of K structures. This is well suited for the example I showed, where you have this ribosome in discrete states. But for a lot of molecules, they exhibit more continuous motions, right? Where like a domain is moving around. And so it's hard to represent those forms of heterogeneity with just a discrete set of structures. Generally also, we have to set K ahead of time, which can be very ad hoc and challenging. If you don't know what's in your data set, you sort of have to sweep or guess the values of K. And there's also optimization tends to be challenging, especially because as you increase K, you dilute the number of particles in each of those classes. And so that makes each of the volumes worse, harder to optimize. Okay, so that's a rundown of sort of classical reconstruction. And those really highlight the core principles, understanding the more modern algorithms that we'll talk about. So in terms of how do people do reconstruction now for these more challenging forms of heterogeneity? So again, we'd really like to tackle cases like this, right, of continuous heterogeneity. We cannot represent such variability with just K volumes. So we really need to learn a full distribution over volumes. Again, in terms of the image formation model, we can modify it again. Now, instead of a single structure, or multiple volumes indexed by k, we're now going to have a full distribution of volumes, which we say v theta zi. Zi is some latent variable which represents the conformation of a particular image. And in terms of continuous heterogeneity, so you can imagine you have a molecule with some domain that undergoes some sort of linear one-dimensional motion. This zi you can think of as some latent variable corresponding to values of that reaction coordinate along which that domain moves. And so this expression is saying for the conformation of a given particle, we plug this into this distribution, which parameterizes a volume given that specific conformation. So again, our goal for reconstruction is going to be to optimize that full volume distribution, v theta, and also jointly on a per particle basis, estimate this representation of its conformation, zi. In general, there are a lot of choices we can take on how to model this problem. And so the multi-class discrete setting that I talked about, you can sort of view where we still have a z there representing the conformation, but z corresponds to a discrete set of class indices. But now we'd like to tackle this case where z belongs to some continuous range of values, which will help us parameterize more continuous motions. And in general, we can have both nonlinear and linear parameterizations. And so there are a set of tools, 3DVA, Rakovar, where the mapping from this conformation to the volume is linear. So it uses techniques like PCA. And then there are also classes of models, for example, using deep learning that have a nonlinear mapping from Z to this volume space. And again, within this nonlinear parameterizations, there are many different design choices you can make, just using neural representations, which I'll talk about next. But you can also encode different assumptions. For example, deformable GMMs or flow fields, where you make sort of assumptions that you have like a single canonical volume that deforms along some coordinate. So essentially, there are many different design choices we can make. I won't get into all of them, but specifically, I'll talk about the principles underlying cryo-DRGN, which is the tool and software package that we work on in our lab. And so to introduce the architecture, first, we can think about how to efficiently parameterize volumes in this new setting, right? So in the homogeneous case, we essentially optimized a volume like this. We had this cubic grid of voxels, right? And we optimized the values of each of those voxels to build up this volume. There's another way we can parameterize the same volume, which is using something called a neural field. And the idea of a neural field is that we're going to implicitly, instead of storing an explicit cubic grid of voxels, we're going to implicitly represent that volume through a function. In this case, the function takes on the form of a neural network. But when I say a function, it's some mapping from a point in 3D space to the value, the density at that space, at that point. And so in particular, instead of, again, explicitly representing every voxel, here, in order to build up a volume, we'd take this function and evaluate it at a bunch of 3D coordinates, to rasterize, voxel by voxel, build up this volume. Some other details. You might wonder, are both of these like equivalent in terms of accuracy? This can be sometimes harder to optimize implicitly, but there are sort of tricks we can add in to make it accurate. In particular, one key insight that Ellen had when developing this was that instead of giving this network, this function, this raw 3D coordinates, we can featureize these 3D coordinates into some higher dimensional space by transforming it with a bunch of sinusoidal functions. And that sort of helps the optimization of these neural fields in representing these volumes. This concept of and this is just illustrating that same point. So there's this experiment where you can do where, like, ignoring reconstruction, you can do this very simple test of, can one of these neural networks just learn a single 2D image? And if you just try to have it learn this image just using those raw 3D coordinates, it turns out that because of the inductive biases in neural networks, you get something very blurry. But then by transforming those coordinates into this higher dimensional sinusoidal space, we're then able to learn these high resolution images. And so in general, this technique of neural, okay, and this is just one more demonstration that again, for pretty high resolution, images of size, 360 by 360, both the explicit voxel-based representation and the neural representation can converge to high resolution.
Is the positional encoding equivalent to a Fourier transform of the point? Essentially, to do a Fourier transform of anything, you take the sinusoidal and cosines at multiple frequencies and just add them up, right?
Right, we're not adding
representing the amplitudes of the components of the Fourier. So it's like a Fourier spectrum instead of a point. Is that a fair way to think about it, or am I missing something?
I think it's not exactly. So it's the same. So yes, you do. It's similar in the sense that, yeah, you're transforming into a set of sinusoids. It's not a direct, I guess, Fourier transformation. We're not adding them. The coefficients for all those Fourier components, there are different choices you can make, but I think by default, we just draw them from a Gaussian. So yeah, the coefficients of all those different sinusoids are sort of random. They don't correspond to the actual Fourier. And this is just a slide saying that this idea of like neural field representations of volumes or images is not a cryo-EM specific thing. And in general, after the development of it, it's seen a use in really a wide range of fields. Just in general, sort of see an understanding robotic planning and other things. Okay. What I just showed was just two different ways of representing the same volume, right? Either explicitly or implicitly through this function. Why is that actually useful? It becomes more useful once we start thinking about heterogeneity, right? And so in particular, when it comes to these explicit voxel-based representations, in order to represent heterogeneity, you need to store and optimize multiple discrete volumes. Whereas in the neural field setting, this is so much more expressive because what we can do to represent heterogeneity is to simply feed in an additional input to the network, which corresponds to that conformation latency, right? So in addition to this 3D coordinate, whether we want this function to represent one structure or 10 structures or 10,000 structures, the size of the network doesn't grow, right? We still have just this same structure of the network. All we change is the value of this conformation latency. And so in this manner, the implicit representation is a much more scalable way of representing multiple volumes corresponding to explicitly storing and optimizing explicit voxel grids. One remaining question is that I've been talking about this conformation latent Z, which represents the conformation of each particle. Where do we actually get that conformation from to feed into this neural field? We do that using a second neural network. And so this cryoDRGN tool for heterogeneous reconstruction, the full architecture is a variational autoencoder consisting of two main parts. The first is an encoder. The encoder takes as input a single one of those 2D images. And based on this, maps it to this conformation latent Z. And then the decoder, which is this neural field we just discussed, takes as input that lane variable and produces a volume corresponding to that specific conformation, which has been estimated from this image. How do we train such a model? So if we consider a single training example, right? So we take our image, we push it through the encoder. This gives us, again, this latent variable Z. By the way, Z typically takes on, we empirically find that between 8 and 16 dimensions works. In general, the Z should be as many dimensions as there are degrees of freedom within the underlying molecular system. So again, image goes to the encoder. You get this lane variable representing that image's conformation, push it through the decoder. And for the sake of training, again, we know based on the Fourier slice theorem that this input image is a centrally oriented slice of the output volume. So what we can do, again, and also recall that the neural field maps a single coordinate in space to the density at that space. So if we know the poses, assume we know the pose for this image, know the orientation, we can evaluate the decoder along each one of the points corresponding to that pose, right? So we know that this image fits into the volume in some specific pose. So we can evaluate the decoder along every point corresponding to the image plane of that image. And then we can take the loss, so just the error, MSE error between the predicted image and the original image. And that sort of provides this training signal, which optimizes the values of the decoder as well as the encoder in sort of this end-to-end fashion. Okay, the main takeaway here, I think, is just that this is sort of a really expressive model, especially for continuous heterogeneity, right? We're not optimizing multiple discrete voxel grids. We're essentially mapping each images to this continuous space of conformations and then using that to decode a continuous distribution of volumes. One last point, just connecting this back to sort of the classical reconstruction. Even though the architecture looks maybe quite different from the EM algorithm we discussed for homogeneous reconstruction, there actually do exist some deep connections, particularly in the EM algorithm, the poses and the class assignments were also latent variables that we sort of did this marginalization over by computing with the full posterior. In the same regard, these conformation variables Z are also a form of latent variables. In general, it's much harder to compute the full posterior over these, right? So for poses, we enumerated a bunch of poses and tried out each one. It's much harder to do for this the conformation variable Z because again, the space is much more high dimensional, maybe 8 or 16 dimensions. It's sort of unbounded. And so we need better ways of sort of predicting that conformation Z, which is why we bring in an encoder. This is sort of the idea of amortized inference, which says we can, instead of like trying out every value of Z, we can sort of reduce the space and find plausible ones by mapping directly from the input image to that value Z. Okay. Finally, in the last couple of minutes, so that's the underlying cryo-DRGN architecture and training. How does this get applied to real data? So first of all, once you've trained this model in the fashion I said, you can encode all your images, again, to get a latent variable for representing the conformation for each one of those images. You can plot them, and so you get a plot like this. Again, each one of the points corresponds to that low dimensional latent representation for one of those images. And so this gives us sort of visualization of the empirical data distribution. And so from here, what we can do is do things like we can sample volumes along continuous trajectories in the space. We can cluster the space, maybe using k-means clustering to get representative discrete samples. And we can also identify sort of these clusters of particles that you can then refine in traditional tools or filter out in the case of Zhong. So a couple of quick examples. Here's this, again, ribosome, a ribosome data set where you have multiple states, discrete states with subunits sort of bound or unbound, depending on where in the maturation process this ribosome is. Traditional processing in the 3D classification-based approach, as I said, leads to procedures that tend to be very iterative and hierarchical. So you run one round of 3D classification to get maybe 5 subsets of particles, then you have to do more rounds of 3D classification on each of those subsets to divide them further, et cetera, and ends up being sort of this iterative process. Whereas with cryo-DRGN, and in general, these more like expressive models of heterogeneity, empirically, these models are able to learn a lot more in just a single shot. And so here, training this model on this data set, each of the major clusters corresponds to one of the major states. But then if you dig deeper within each of those clusters, you're also able to pull out all those minor states all in just like a single round of training. And also additionally identify sort of these rare states. Again, because the process isn't super ad hoc and iterative, you're really able to pull the most out of your data. One more example, So this is a spliceosome. So the previous example showed how even for a case of discrete heterogeneity, these neural models are really expressive, but they're especially expressive when it comes to these more continuous forms of heterogeneity. Here's the spliceosome. Traditionally, and what you do to process this is manually identify different domains, different moving parts, and reconstruct and refine each of those separately. This is showing that, again, sort of in a single shot without any strong priors on what domains are moving, you can train one of these models, and sampling these continuous trajectories, in this case, the principal component of the lane space, allows us to visualize these more continuous dynamics. Right, and this is just showing, again, you really get sort of this full distribution corresponding to your entire data set. So each one of the particles in your data sets maps to sort of this unique volume, allowing you to really fully explore the conformational landscape. Okay, so to summarize, cryo-EM is very powerful experimental technique, especially when it comes to dynamic proteins, because this freezing process captures them in this near native state, this sort of equilibrium distribution. We talked about sort of the probabilistic techniques underlying classical reconstruction, which provide the core principles, which then motivate this new class of heterogeneous reconstruction methods, which has emerged over the past few years. And again, these heterogeneous reconstruction methods, you can make many different design choices and code many different priors. I specifically presented cryo-DRGN, which is one particular model which uses this implicit neural representation to provide this really expressive and sort of assumption-free model of heterogeneity.
So tough act to follow. Rish gave an amazing overview of the kind of fundamental theoretical aspects of cryo reconstruction. Hopefully I'll give like broader big picture overview of some of the things that our group works on. So kind of at a high level, the motivation of the methods that our group develops are to develop computational methods to push forward scientific discovery. And so definitely something that we have been talking about, and what I will talk about today, is how we can develop methods for cryo-EM and cryo-ET data analysis, in particular extracting the conformational ensembles of these macromolecular machines in situ. Something that's particularly exciting, especially right now in this landscape of like large kind of deep learning models, is connecting these kind of large scale generative models with experimental data. So that's definitely a major focus, especially for molecular and structural biology. And then finally, I won't talk about this today, but something we're very excited about is developing new AI methods for small molecules and natural products. Okay, there's a couple of repeats, but I'll go through a lot of the intro very quickly. The biological motivation, we want to solve protein structures, and we want to be able to visualize their conformational ensembles, because that gets us much closer to understanding the function of these macromolecular machines. And you might be very aware of breakthroughs in machine learning methods for structural biology, namely the development of AlphaFold and related systems for predicting protein structures. There's also been a lot of really interesting methods kind of connecting these structure prediction models with protein language models, with generative models to do de novo protein generation, like chroma, great alphanumerical proteins right there. And so one of the main kind of takeaways is that these unified machine learning frameworks are state of the art for a lot of kind of tasks in protein structure space. But for a lot of the things that I think we care about as biologists, so putting our kind of structural biologists hat on, I think the goalposts have just advanced. So we're still very data limited for many problems, including kind of understanding the downstream function and understanding kind of how these molecules interact in situ, which is why I'm very kind of grateful that our group works on these methods for advancing the type of data that we can get, hopefully even help with training the next generation versions of these structure prediction data. As Rish already very nicely introduced, proteins and other biomolecules form these large dynamic macromolecular machines. And if we really want to understand kind of the function of these machines, it would be amazing to be able to visualize the entire conformational ensembles, either at kind of like high resolutions or even at kind of broader in context environments. Unfortunately, techniques to study these molecular motions especially at kind of atomic level resolutions, are rather limited. So we have NMR, which is typically limited to smaller proteins. What I like to call kind of avant-garde crystallography-based approaches like EFX, XPEL crystallography. This still requires sample crystallization, but you can maybe look at some of the mechanical deformations of these of the kind of proteins within the crystal lattice. And then in a previous life, I worked on molecular dynamic simulations. So can we actually kind of run long timescale MD simulations and get an in silico microscope into the dynamics of these systems? Very, very challenging to do computationally. And there's also a lot of fundamental kind of challenges in the underlying accuracy of these force fields. Hacking AlphaFold, I think, is pretty interesting. What kind of signals are in the underlying MSAs? But then when I learned about cryo-EM, kind of near the beginning of my PhD, I was like, this is a very, very cool experimental technique that can allow us potentially to extract the kind of conformational ensembles of these nanometer scale machines. So I won't go over kind of reconstruction fundamentals because we just had an amazing primer by Rish. But again, we have a sample flash frozen. We have a collected set of micrographs. And then from there, we have a collected set of single particle images that are characteristically very noisy. And the name of the game is to reconstruct in 3D the electron scattering potential, or this kind of volume function, from our collected set of images. And this I definitely won't go over since we just had a primer on it. The goal is to estimate our volumes and the poses typically with maximum likelihood techniques. And there's a lot of state-of-the-art software packages for doing this, especially in the homogeneous case. So Relion, CryoSpark, Warp, Freeline, EMAN, and so on. Rish also gave a quick overview about this, but I think this provides a nice intuition for this iterative EM algorithm that we talked about. And so this is the Fourier slice theorem. The Fourier slice theorem states that the Fourier transform of a 2D projection of a volume is a central slice out of the 3D Fourier transform of the volume, where the orientation of that slice is perpendicular to the projected direction. And so if we have a set of images and we know their poses, we can convert or take the FFTs and then insert them as slices into the correct kind of orientations in 3D space, and then take the inverse FFT to get back our real space kind of visualization of the volume. And then that kind of gives the intuition for how these iterative EM algorithms work. Given some initial volume V, you estimate your poses. Then given those poses, you re-estimate the volume and rinse and repeat until convergence, ideally to the global minimum, although there's a lot of challenges with that. And Rish already gave an overview of these state-of-the-art software packages for doing that, namely Relion and okay, so that was homogeneous reconstruction, but we have this heterogeneity problem, which is the kind of major opportunity we're interested in. So each cryo image contains a unique molecule and potentially a different conformational state. So the ability to image these heterogeneous structures is a major opportunity in cryo-EM, and especially relative to other techniques like crystallography, where we've experimentally rigidified each copy of our molecule in the exact same conformation. In cryo-EM, we actually get the experimental data needed to study the kind of intrinsic structural landscape of these protein complexes. Unfortunately, dealing with this heterogeneity is very challenging, and standard approaches really include just ad hoc filtering or getting rid of as much of it as possible, which is obviously not ideal if you're interested in intrinsic heterogeneity. And then something called multi-class reconstruction, which we already actually talked a lot about. And that leads to these kind of very manually made SI figures in all the cryo-EM papers, where a practitioner has the expertise to know like how to sweep, how to choose subsets, how to combine them. And the main takeaway here is that at the top of this processing pipeline, there are millions and millions of images. But by the end, you have two, albeit high resolution structures, but you've also thrown away a significant portion of your data. So in this case, maybe 90% of the images. The question is, what's in that 90%? And so the identification and analysis of heterogeneity, especially continuous forms of heterogeneity, is an open problem in cryo-reconstruction. And this is a problem that I originally sought to kind of answer in the beginning of my PhD. So can we design a modern machine learning method for this heterogeneity problem in cryo-EM, and still continues to be kind of a major research question in my group. Okay. So you'll also kind of be familiar with this slide. So in heterogeneous reconstruction, we extend the image formation model. Instead of a single volume for each image, we now have this distribution of volumes parameterized by some latent variable model. And I think it's important to take a step back and think about what are the sources of heterogeneity, because that motivates kind of how we design these, how we want to design our volume distribution, our parameterization of the distribution. And so, yeah, I like these acronyms because it becomes a mouthful to say conformational heterogeneity and compositional heterogeneity over and over again. But we have conf-het, which can originate from shape deformations of our proteins, our biomolecules, and then we have comp-het, which refers to kind of compositional differences in the sample. So you can think of that as like object class. And then in practice, there's usually a lot of real het, which comes from artifacts and non-idealities during imaging, maybe crystalline ice forming on some of the images that get falsely picked during the particle picking. And different methods, there's now a lot of different methods that use different choices of the volume parameterization, latent variable model, and also the way they do inference of all these different latent variables. And so there's a lot of different design choices. And then one last thing to note is that most methods treat poses as known before doing heterogeneity. Okay, so that's the setting of like heterogeneous reconstruction, cryo-EM. Our method, which is called cryoDRGN, it's an acronym. It stands for Deep Reconstructing Generative Networks. And here our task is 3D reconstruction. Instead of a single kind of homogeneous structure from our data set of unlabeled images, we want to reconstruct this low dimensional continuous distribution structures. So yeah, Rish already gave an overview, but we frame this heterogeneous reconstruction problem as unsupervised learning of a deep generative model, where the generative model is parameterized using this neural field. And it's particularly exciting to see kind of the broad applicability of this class of methods in computer vision. And fundamentally moving forward, we're interested in developing new methods that kind of broaden the scope of what we can get out of cryo-EM data and structural biology. Okay, so I'm going to skip over these real quick, but the main takeaway that Rish already nicely went through is that cryo-DRGN structures are parameterized using a neural network instead of a voxel array. And then the main reason we do this is that it gives us a very elegant way of modeling, especially continuous forms of heterogeneity. So instead of having to model k separate voxel arrays, again, there becomes this bias variance trade-off, especially with larger numbers of k, as well as computational considerations. We can simply extend the input dimension of our neural network to condition on an additional latent variable vector. Okay, this slide we're also already familiar with. And so I don't think I have anything else to say other than it's all expressed in Fourier space. So each input image is a 2D planar slice out of the volume in the Fourier domain, even though this image is kind of space. Okay, and the main advantage is that this is a very flexible model with very few initial assumptions about the data. And so this kind of neural network or this coordinate-based neural network, whatever kind of distribution of structures it can model, or it can learn from the data we can model. Okay. So at test time, we have this encoder and decoder. And again, Rish already went through this, but given a set of images, we can encode them into this low dimensional latent space and just directly visualize that space, see if there's anything interesting. We can even learn about maybe the clustering and this empirical data distribution. And we can use the decoder neural network to generate sample volumes at different values of our latent variable. So we can view representative samples. Maybe given two end states of interest, we can do an interpolation in the latent space or maybe a random walk in the latent space and generate the corresponding kind of continuous trajectory, continuous model. And then we can take subsets of our data set to either filter from a lot of kind of real artifacts or validate kind of an individual structure using traditional kind of homogeneous.
Quick question. So you mentioned that this approach uses none or very few assumptions. Mm-hmm. So what are the assumptions it uses and how does it compare with the assumptions made in the traditional approach where you assume it's sampled from a normal distribution?
Yeah. I guess the main assumption is that the distribution of structures can be represented in some lower dimensional space, like some e-dimensional space. But we don't make any kind of more strict assumptions about what that distribution might look like. Cool. So in the original development of cryo-DRGN, we did a lot of validation on synthetic data sets just to make sure that everything was working. But then something that I was very kind of motivated to do is to create an actual software package around this tool that people structural biologists and cryo-electron microscopists can train these models to analyze their data sets and visualize the heterogeneity. And so Rish already went through these two examples, but we have this kind of assembling bacterial ribosome where we're able to discover new structures. And then this precatalytic spliceosome on the right, where we're able to visualize these kind of large-scale continuous dynamics of this large macromolecular machine. At Princeton, I'm very lucky to have Michael in the group, who is a research software engineer who has taken over like maintenance and support of this software package. That's been absolutely amazing. Hopefully, it's relatively easy to use with a pretty straightforward software pipeline. We have extensive tutorials, maybe too much, but we'll see on how to use the tool and how to kind of interpret the results. And we're working on kind of enhancements and integrations, some of these new kind of pose estimation methods, getting that kind of integrated into the main project. Cool. And so there's a lot of like kind of interesting things we can do with cryoDRGN, but there's all the limitations which have motivated the kind of new directions in cryo reconstruction that our group is currently working on. So I will just give a quick overview of a couple of these different methods, but I'll focus on cryoDRGN ET, which is how we do structure determinations. Okay, so I think it was great to get in the primer a deep dive into this pose estimation problem, because it's actually very, very interesting and very engineering intensive and usually just kind of completely skipped over. But to obtain kind of this rendered slice by the Fourier slice theorem, you need to know the orientation of that slice. So we have this pose, which is a tuple of the rotation, and a 2D translation. So we need some model to approximate the pose, or we need some approach to approximate its pose. How do we do this? I won't go into too much detail, but a lot of the research in the group so far has been kind of exploring different ways for doing pose estimation. And this is a setting of cryo-EM called ab initio reconstruction. And so that's where we start reconstruction without any prior estimates of the poses. We've talked about exhaustive search, so this hierarchical kind of grid search, which is very robust to high noise levels because we're kind of exhaustively enumerating the whole space and testing them all. It's very slow. So we're not doing 19 million slices, but we're doing, thousands of slices. And especially with a neural model where we have to do a full forward pass through a neural network, that's very slow. Our loss function is differentiable with respect to our pose variables. So we can, just do direct gradient descent on our poses. That's very fast, but this requires initialization. And then we've also explored various kind of amortized inference approaches, or basically put on our deep learning hats and been like, let's just train a model to predict the pose given the input image. This is very fast to evaluate, but this is also kind of unstable to train. It needs learning and is tricky to show in kind of this truly kind of generalizable setting. Okay, so we've tried all the above, and just skipping to the punchline. The latest version, which we've called cryoDRGN AI, incorporates the exhaustive search and this SGD, or gradient descent-based approach to pose estimation. And really the research question is, can we perform single-shot ab initio reconstruction of modern cryo-EM data sets? They're very large data sets that have millions of cryo-EM images. They're typically unfiltered, so there's kind of these outliers in the distribution, and do this in like a reasonable amount of time, so in kind of a scalable fashion. I won't go into detail, but we switch off between this hierarchical pose search and an autodecoder framework. We address kind of, the trade-offs between memorization of the input images versus generalization. There is regularization from the neural model, and we've been able to show the discovery of rare states from very large kind of cryo data sets. And this is driven by Axel, Rish, and Brian. There's a couple of directions that we've been extending our cryoDRGN AI method. And just really quickly, one is modeling complex mixtures using a kind of mixture of neural fields. So we call this model Hydra. And kind of one direction we're pushing is, instead of, a single neural field, can we have like what is a bottleneck if we really want to reconstruct a complex mixture or complex sample? So this is kind of this hybrid architecture combining cryoDRGN AI and 3D classification. And we're doing both inference of kind of which neural field, kind of which class we're in, as well as the conformational state and the poses. So it's a very, very challenging inverse problem. here's some results on a synthetic data set. We mixed together moving ribosome, moving spliceosome, and moving codon spike protein. And we can reconstruct all three classes simultaneously without any labels of the unknown latent variables. And then applied to a real data set of red blood cell lysate, we can recover both this RIR complex and a couple of other complexes present in the data set. This is kind of doing the hierarchical classification-based approach with CryoSpark. And in contrast, just using a single neural field or cryoDRGN AI, we get one class that looks like the RIR complex, but then the other class just becomes, or the other region in the latent space, sorry, it's just one class, becomes a jumble of the other kind of junk. That's one extension of this ab initio approach. The second is moving into cryo-ET. And so I'll focus a little bit on this on the kind of last end of the talk, but I'll just mention that instead of doing cryo-EM, or instead of having a single image per particle, now we have a series of images. And we adapt this two-stage post-estimation procedure. This is really driven by Rish to the ab initio setting for subtomograms in situ. And then just showing one result slide. This is the kind of translation elongation states of the 70S ribosome from in situ data, fully trained, fully ab initio. Okay, so we're making progress on ab initio reconstruction. One thing to note is that all these kind of reconstruction methods we've talked about are purely image data driven. So we don't kind of leverage any information we have about the underlying kind of chemistry or the sequence of the molecules. So one, I think, direction that's very interesting, and I think a lot of people are working on it right now, is can we bridge these methods between protein structure prediction, we're using, the sequence information, all the evolutionary related sequences, and protein structure determination, where we're taking kind of imaging data and doing this inverse problem, analyzing the measurement data. Cryo reconstruction methods currently don't leverage any of this prior information about the underlying protein chemistry or physics. But if we did, could we, push forward what we can get out of the data? Yeah.
On a related note, it's not really directly about the, protein language model to, or the sequence to structure, but it seems to me that at least the two types of images that Rish showed, in the end, there was a ribosome. In the beginning, it was a different protein. Those looked very different. So it occurs to me that one may be able to classify these 2D cropped images as this protein, even if we don't know what the conformation is, and maybe do some sort of supervision on
I think that would be really interesting because I think this purely unsupervised way is just going to get harder and harder with more and more complex samples. And we do know a lot of stuff. Maybe we don't even need the sequence. We just need like templates or something like that. So I think that would definitely be a really interesting direction.
But it's not really done yet. No, okay.
Good research. Cool. Okay. So I don't have anything super kind of groundbreaking, but one kind of proof of concept direction that we've been taking is to use these pre-trained protein diffusion models as a prior in various types of inverse problems in protein space. And so we're not tackling 3D reconstruction yet, but we've been doing some toy experiments where we're filling in we're doing like protein infilling, satisfaction of NMR distance restraints, and cryo-EM atomic model building. And here what we're doing is turning partial and noisy measurements from our experimental data and combining this with a pre-trained diffusion model. Here, Chrome. One thing to kind of keep in mind that is always top of mind, especially when we're combining these maybe like large generative models trained on existing proteins versus new experimental data of new proteins that we want to solve, is how much do these kind of pre-trained models limit the ability to discover new things? So I think that's like one, maybe just like core question to ask and core question to consider, especially with respect to validation and just with respect to what do you want to get out of your experimental data. And so that, led to kind of this project on benchmarking, right? So now with these kind of large protein generative models, maybe with some of these other reconstruction methods that use more, like opinionated forward models, how do we actually interpret the results, especially when we're considering heterogeneity? So we're in an era now where reconstruction of these molecular movies is possible. You get these like really compelling and beautiful visualizations of these macromolecular machines. So here's like a couple different methods. The representation, so like how we actually model this distribution of volumes matters. It really can potentially, it does bias the results of the reconstruction, and it should really depend on kind of what prior information we have about the system and also what biological question we want to answer. One kind of unfortunate, truth is that there's no ground truth for real data. So we don't really know what's in the data. And evaluation, especially when we're kind of comparing these different models, currently requires just benchmarking by eye. So like, does this look reasonable? Does this match our priors? And so in CryoBench, this was led by Minkyu, a graduate student in the group. We built a large data set, a large synthetic data set, modeling different types of heterogeneity. So I'll go over that briefly. And then we benchmarked 10 different methods, both deep learning based, linear, nonlinear ab initio methods, and also came up with new metrics and ways of comparing these different heterogeneous reconstruction methods. One thing that I think is particularly useful for methods developers is the different kinds of data sets that we developed. So we have both kind of easier versions of both ConfHet and CompHet that are useful for diagnostic purposes. So this IgG1D is an IgG antibody that has this simple one-dimensional circular motion. And so what you're looking for in the latent space is a nice rainbow circle. And also riboassembly, which is a mixture of ribosomes, so very canonical cryo-EM. And then we are also considering, we designed very challenging data sets where no methods currently perform that well to motivate kind of new methods development. So one is IgG-RL. So here we're kind of sampling random dihedral angles from this linker region. And so I think this is for an antibody, IgG antibody, and the Fab is oriented randomly. But I think that's representative of a lot of systems where we have a smaller domain that's kind of moving and is kind of disordered relative to a larger fixed domain. SpikeMD is a data set generated from a long time scale molecular dynamics simulation. So instead of just like 100 underlying ground truth structures, in this data set, there's 46,000 underlying ground truth structures. And so an interesting question is how well does do the methods really approximate this much kind of higher dimensional space of structures? And then finally, TOMO2 is a mixture of 100 different complexes commonly found within cells. So instead of sticking to 4 different ribosome assembly states or a couple that you usually get from these structural biology papers, can we push the envelope in terms of much, much larger sets of protein complexes? Okay, so cryo-EM, we still definitely want to check the results qualitatively. So here's, I think, cryoDRGN results on these different data sets. So again, IgG1D, we have a nice rainbow circle. Riboassembly, we have like 16 different clusters. And maybe in these cases for SpikeMD and TOMO2 it becomes less clear how we interpret the results. And especially when we want to compare between methods, right? And so here's cryoDRGN and a bunch of other heterogeneous reconstruction methods. We have both the structures and also these latent spaces. And so, yeah, like how do we analyze them? Yeah.
So for like MD Spike, in this situation, like in the previous slide, it seemed like there were a contiguous distribution of states. So would we expect the UMAPs over here to be kind of as like, would something doing well, would we be able to qualitatively tell by saying like, oh, the UMAPs look relatively continuous in this case?
Yes and no, like you would think, right? Like if you have a continuous distribution, you would want a continuous UMAP, but then the layout of these latent spaces is very arbitrary. So how do you actually, quantify the results or like quantify how well these distributions are learned and what can you really interpret from these UMAPs? Yeah, very inspired by similar challenges from the single cell literature, for example. And so one nice thing is that we do have quantifications because this is synthetic data where we know all of the latent variables, all the unknown variables that are hidden from the reconstruction methods, we can kind of quantify these volume reconstruction metrics. One thing that we note is no method dominates across the different forms of heterogeneity. So like ConfHet, and types of like conformational heterogeneity. ab initio reconstruction methods are still needed. So this like joint conformational inference and pose estimation is very, very challenging inverse problem. And these metrics, they work. You can kind of compare, you can make these large tables, which I think people in machine learning really like. But better metrics are still needed. And I think one interesting research direction, especially if you come from both the kind of ML and the structural biology background, is designing better metrics, maybe that are data set specific, that can really answer kind of deeper questions about, you know, the different conformational states and microstates, for example, of the molecular dynamics. Okay. So what is this all working towards? I've talked about ab initio reconstruction. I've talked about kind of benchmarking. And really the point of all this, of this heterogeneous reconstruction, is to move to, in my opinion, is to move to more complex samples. So instead of like solving a single structure in its conformational state, can we instead solve complex mixtures or even structures from intact cells? Okay. So now we can kind of talk about cryo-electron tomography. I will just do that by showing this. Instead of a purified solution of a molecule, a purified solution of your protein or biomolecular complex of interest, here you have a whole cell. A whole cell is usually too big to be electron transparent, depending on the cell. And so what you do is focused ion beam milling. You use a beam of ions to mill away, kind of ablate the top and bottom. And so what you end up with is a very thin cross section of your cell. And then instead of a single image, now we do tomography. So it's the same electron microscope, it's the same transmission electron microscope, but instead of just a single image, now we spread our electron dose across a series of images, in practice, 41. So we take kind of 41 images as we rotate this imaging stage, and we see this 3D kind of slice out of the cell in all these different orientations. So this is a tomogram on the right. Yep.
So the ion beam milling is literally physically manipulating the cell. Is there any concern over that's affecting the conformation that the proteins are in, or disassembling?
Yeah, so
certain
it's already
pieces?
frozen beforehand, but there is a lot of kind of like sample preparation considerations. So if it's a large enough cell, you want to high pressure freeze it so it all vitrifies at the same time. And then this is actually kind of beyond my area of expertise, but I think there's like a plasma focused ion beam mill is less likely to deposit a bunch of other stuff than like a metal ion beam mill or something like that. So there are, you know, Thermo Fisher is working on it and interested in people buying their stuff. Okay, and on the right, this is a tomogram. So this is, we take the like 41 separate images or the micrographs and then just do a 3D reconstruction. And we can visualize here, you know, a bag of ribosomes. This is a service. And then I think what's been so exciting about cryo-ET is that even though we're looking at things at low resolution, we can computationally kind of do the same trick that we do in single particle cryo-EM. So we can take individual instances of our molecule of interest and computationally like orient them, combine the information, aggregate the signal, and get near atomic resolution structures. But instead of, you know, a structure of your protein in a kind of crystalline lattice, instead of the structure in kind of aqueous solution in vitro, this is the structure of the protein complex, the ribosome here in situ. So the actual native structure that's closest to the functional state of the molecule of interest. And in this case, they were even able to see the structure of a small molecule antibiotic bound. Okay, amazing. So why don't we do this for everything? You know, unfortunately, the cryo-ET processing workflow in practice looks something like this. So this is literally the workflow that I think Sagar did to process some of our cryo-ET data analysis. You can kind of appreciate that the metadata is a lot more complicated now because we have this set of images, 41 different tilts. You need to align them all in 3D, and like, you know, how much you move the imaging stage, but obviously you want to like re-estimate that, and then you have to like pick out the subtomograms, and then there's actually all these other considerations on whether we take like 3D data versus 2D data, and it just becomes a nightmare practice. It's all academic software as well, so yeah, I'll just stop there. And in the cryoDRGN ET project, or I guess one more point I want to make about this kind of cryo-ET processing workflow is a lot of the, repetitive nature of it is because of the heterogeneity, because the particle picking problem is not solved, although there's a recent Kaggle challenge about that, spearheaded by the Zuckerberg Imaging Institute. And so you're picking a bunch of like random stuff that's not necessarily your protein of interest. And then you are computationally trying to filter it and analyze the heterogeneity and kind of classify the different states. And it's very, very slow. And yeah, that's the heterogeneity is definitely one significant contribution to why this processing workflow is a bit of a nightmare. And so something that we worked on in the group right when I started is extending cryoDRGN, which is ideally, like in the ideal case, useful for single shot heterogeneity analysis of single particle data to cryo-ET data. So the high level kind of schematic looks pretty similar. And so the main changes are, instead of a single view encoder, we do a multi-view encoder. Actually, this is a little bit different than the standard way. Subtomograms, which are kind of these subvolumes, are analyzed. So we actually take the individual tilted images. And then the forward model, instead of single particle cryo-EM, is cryo-ET, which is very similar. We just have to take into account kind of the details about like electron dose exposure. Yeah. And then kind of just highlighting in single particle, we have a single image, but in subtomogram averaging, we have kind of multiple views, which is actually kind of interesting from a hierarchical standpoint. And in the kind of development and analysis of cryoDRGN ET, we were able to apply it to the same 70S ribosome from the Mycoplasma pneumoniae data set done by the Mohammed Lab at EMBL Heidelberg, and see the same kind of translational states. And also in collaboration with Thermo Fisher, they collected this very large data set of plasma fib-milled lamella of S. cerevisiae, and we were able to characterize the conformational landscapes of the ribosomes and fatty acid complexes. Okay, I think I'm actually going to go through the cryoDRGN ET results kind of quickly just to save time for questions. But again, just the overall pipeline, we have a very lamella, we reconstruct a 3D tomogram, and then we pick the individual instances, solve their structures, get the conformational landscapes, and then in addition to seeing the conformational states, we can also map them back in spatial context. So I think that's a really powerful aspect of cryoDRGN ET. This is what we get out of cryoDRGN ET. So here's latent mixed space representation. In this case, maybe like 130,000 particles of the ribosome, or, sorry, 119,000 particles. And just by visual inspection, we had one cluster that was just like kind of Zhong particles, one cluster that looked like the ribosome in the rotated state, and one class that looked like the ribosome where the small subunit was in the non-rotated state. Because we don't want our model to be wasting its expressive capacity on these bad particles, we just filter the data set with the good particles, create it again, and we again see this nice kind of partitioning of the two rotational states. And then we dug deeper. So we're not just looking into the two like ratcheted and not ratcheted state. We just like visualized and mapped out all these different elongation factors, initiation factors, and also the actual tRNA binding site. And a video of that is shown on the right. So these, especially if you're a maybe crystallographer, this is like very blobby, right? So you don't like see the high resolution details. But then what we did in this case is we used cryoDRGN to kind of classify these different states. And then we can do the traditional back projection using the full resolution images, higher resolution structures of these translation elongation states. As another kind of demonstration, instead of the ribosomes, which are very abundant, we also analyzed the fatty acid synthase complex. So the traditional kind of subtomogram averaging approach from the picked particles looks something like this. when we look at the same data set with cryoDRGN ET, we get this latent space and we see kind of a distribution of structures, only one region of which actually looks like the fatty acid synthase complex. So when we take this subset of particles and then do a refinement, a traditional refinement, we now get a much higher resolution, beautiful structure of the yeast fatty acid synthase. On the left is showing kind of the training progression of a cryoDRGN ET model on just like on the full data set. And I think one of the main takeaways was that this happens on like less than an hour. So like in only a few minutes, we can actually distinguish good from bad, which would be really good for this kind of very slow classification process that takes like weeks or longer. We run a new model on just this subset of around 5,000 like quote unquote bona fide particles. We actually begin to see the very subtle rotations of this fatty acid synthase complex between the two known states. Kind of big picture, so we can visualize these interesting intermolecular conformations. One thing that's interesting is integrating this processing pipeline with the traditional processing pipeline. Something that Rish has been working on recently is the ab initio reconstruction in cryo-ET, where we have like much more complex samples. Since this is the actual kind of collection of protein complexes that you see in vivo, in situ. And so can we actually process this in silico to reconstruct molecular landscapes? This is a slide from my Thermo Fisher collaborator, although I think he's recently moved to St. Jude. Traditionally in structural biology, the analysis of data sets is very kind of like done by the group. So each group will analyze, collect a data set, analyze it, and deposit it into the PDB. Cryo-ET data kind of fundamentally breaks the paradigm, I think, of structural biology, because in a couple of ways. One, it's like much larger in scale. It's much more expensive kind of experimentally to collect the data, extremely expensive computationally to process the data. It's very, very hard. And the data set doesn't just give you a single structure. It gets you kind of the entire molecular details of the whole organism. And so I think there's this new paradigm that especially folks at the Chan Zuckerberg Imaging Institute are really kind of inspired to do, which is instead of all kind of working on different things, all collaborating together and analyzing as a community. Cool. And so to summarize, again, just recapitulating some of the things that Rish said. So cryo-EM and cryo-ET are particularly interesting modalities since we capture this new type of data of where molecules are in a quote near native state sampled from a quote equilibrium distribution conformations, where these quotation marks are the caveats due to sample kind of preparation. There are now many tools, in addition to cryoDRGN, becoming available to study heterogeneity in cryo-EM. So as a practitioner, kind of the choice in algorithm encodes a lot of different assumptions about the heterogeneity that you might visualize from the model. And so that's always important to keep in mind. That's driven our benchmarking work. cryoDRGN's kind of neural field is a very expressive model of heterogeneity. And so I think that's nice to be able to discover new states in a relatively unbiased setting, as well as visualize these continuous dynamics. And then the last thing I want to end on is a couple of thoughts of cryo-EM in this post-AlphaFold era. Or not. There we go. So what's next? I definitely think there are very interesting things to do in just this machine learning for cryo reconstruction setting for heterogeneity. How do we bring in the kind of underlying atomic models or the underlying chemistry or physics? But I do think kind of models that break this paradigm will be very kind of beneficial for the whole target processing pipeline. We can really broaden the scope to more challenging targets or challenging systems. Cryo-EM right now is still, quite bespoke and manually intensive, especially this traditional 3D classification procedure. So can we, commoditize and just like automate a bunch of the cryo-EM processing? And can kind of new machine learning methods motivate different paradigms for imaging and processing, right? So are we really stuck to just like single image, and can we design different ways of collecting the data for heterogeneity? Obviously, moving forward, kind of the one kind of main takeaway I have of especially cryo-EM data is that these current generation of structure prediction models don't predict dynamics that well, like the different conformational ensembles. And so if we can just collect the data to train the next generation of prediction or design algorithms, I think that would be very exciting. So closing the loop between prediction and determination and really kind of unlocking new applications in kind of understanding protein dynamics, protein design, and also the interaction with other types of molecules that are not as commonly found. And then finally, bringing this all together for in situ visual proteomics with technologies like cryo-ET, as well as other types of biochemical and biophysical. And with that, thank you all for listening to 2 hours of cryo-EM, almost. And I would especially like to thank members of my group. So Ramya and Ryan drove the cryo-DRGN ET project. Axel has been doing a ton of work on both pose estimation as well as our APD 3D method, which with the chroma or the protein fusion models. Rish has played an instrumental role in like so many of these different projects. Minkyu drove CryoBench, and Michael has been supporting the software and integrating a bunch of these new methods into cryo-DRGN, as well as like to thank the other members of my group, a bunch of our collaborators, and our funding sources. And thank you all for listening.